All right, welcome back, students. Rishi is going to kick us off today with some more info about the final project and some announcements about some our competition results. Take it
Oh,
away, Rishi.
actually, wait, just like I have to leave and rejoin. Sorry.
No worries. Okay.
My yeah, I have to be able to share screen. My laptop's having some issue.
Okay.
Can everyone see this? Cool. So yeah, so we're getting to the kind of part of the semester where we start talking about the final project, and for all of you guys to start thinking about what you want to work on, and what sort of project you want to do. So we were thinking today, I'm just going to sort of walk through some of the general types of projects students have done in the past, sort of broad categories, and then also just walk through a couple concrete examples, just to give you all sort of an idea of what's possible, what you can do. But nothing I go over here is like exhaustive. If you have some other ideas beyond this, feel free to explore it, run it by the TAs, and whoever. Okay, so here we go. Uh, so yeah, so first, just kind of talking about overall general types of projects. One of them obviously is sort of a rendering project you could do. So this would kind of follow in the path tracer projects, so projects 3.1 and 3.2 mainly. And then it would mainly be sort of extensions to those. So one of the things you could do is sort of volumetric scattering, where you're rendering a scene where there's something. Basically, it's not just air. We have smoke or fluids. You're sort of rendering a little bit more complex scene. The rays maybe are not guaranteed to be straight, things like that. So a little bit of complications there. You could also do something like photon mapping for caustics. So photon mapping is a kind of slightly different rendering technique to like ray tracing and path tracing that we've been learning about, that allows us to sort of render some more artifacts, like light artifacts, some more complex things. Um, so that's sort of an interesting add-on. And then another thing you could do is sort of adding textures to the current path tracers. So right now we only sort of deal with materials, diffuse materials, specular, whatever. But adding textures could sort of be interesting and increase the like possibility of scenes we can render using our path tracer. So rendering sort of one, broad category. And, like I mentioned, there's a lot of other stuff you can do in rendering beyond just what I mentioned here. Then another one is geometry. So this is sort of following on project 2, if you sort of really like that or, enjoyed it. So one of the things you could do is like mesh simplification. So this is, in the project we did subdivision, sort of making a mesh more fine, more detailed. This is sort of the opposite, where we reduce the number of triangles while still sort of trying to keep the shape relatively the same, and see, how many triangles we can take away, things like that. Also, you could do remeshing. So we briefly talked about in lecture, sort of, what makes a good mesh, whether, some types of triangles are sort of more preferred than others. So whether we can, change meshes into better versions. And then another thing, sort of point cloud to mesh conversion. So if you have a bunch of points in 3D space, can you kind of create a mesh out of it? Maybe for some use that's better there, like rendering. So, lots of other things here with geometry, you know, um, other things you can explore beyond just this, beyond meshes, and other data structures. Okay. Um, and then another big one is simulation. And we're sort of just entering this phase of the class right now. With this last discussion, rope simulation, we're going to do a lot of clot simulation for project 4. And yeah, so simulation is another super interesting topic, and kind of a favorite, because you get some really nice looking results and it's kind of fun to play with. You can do a lot of interesting stuff. So one of them is obviously you could do a smoke simulation. Lots of students and projects have done this. You could do water simulations, snow simulations. In the past, students have done sort of terrain simulations and then simulating water flowing over it. So you can also sort of expand outwards and make kind of more complicated add-ons to these simulations. Like some groups have done like smoke simulations that respond to music playing and things like that. So lots of interesting stuff to do in the simulation area also. And yeah, here's just another picture, one I thought was really cool from past project, just sort of a ferrofluid simulation. So a magnetic fluid, so kind of more interesting, but just an example of add-ons you can do and how you can make it more interesting. And then another sort of area that you can definitely talk about is imaging or computational photography. So we talked a little bit about this with lenses. If you're doing sort of the depth of field part of this project 3.2, you may have had a little bit of experience with this. But so this is kind of looking at different types of cameras. So light field cameras or something we'll learn about, I think, in a week or two, maybe, where you can essentially refocus an image after you've taken it. So building tools that allow you to refocus images. Another thing you could do is sort of multi-shot HDR. So taking different pictures with different sort of exposures and all, and then putting them together to get one picture that has a huge sort of range and make it look even better. So some interesting things you can do with imaging and computational photography. And like the others, you can also combine this just like we sort of did for the depth of field and rendering. You could also sort of do other combinations like that. And lots of interesting options here. Okay. And then the last one, so one of the topics sort of, students have done in the past is games. So this is just an example of a game someone did last time where they sort of brought a lot of the geometry, physical simulation, rendering topics we talked about in class and sort of put them together to create an action game that they could play and run in real time. And it was a lot of fun. In the past, students have worked on sort of VR games with special sort of rendering effects, and lots of interesting things can also be done here while also having some fun. So games could be cool. Okay. And so, yeah, so those were kind of some of the overarching topics, sort of the different sections of this class that we've covered and, could be good to have final projects in. And so now I'm just going to talk through some of the sort of, concrete or examples of projects in these different areas. And yeah, once again, the stuff I've said in the past is not at all exhaustive. If you have some other ideas beyond anything I just mentioned, that, can totally work. So some past projects. Yeah, so here's a past project. So a black hole ray marching, essentially sort of, seeing how light interacts with black holes and simulating this and creating pictures. And you get this project came up with some really, really cool results. And it looks really fun and it's really fun to play with. They actually have this live demo. Yeah. So here we go. You actually have a live demo. And you can sort of, as you move the black hole around, you see how the light gets distorted by it. And then, they have settings to change the diameter and stuff. But so, really cool project, some nice results, and it's fun to play with. Okay, so that's black hole ray marching. Another one, smoke simulation. We talked about this earlier. This is sort of an example of it. So I'll let it play. Yeah, so basically as they click, smoke kind of gets added into the scene, and then they have a bunch of potential obstacles, so you can kind of see how the smoke responds to them. You can change different kind of properties of the smoke. You can see different properties of the smoke, change the color, things like that. So a really good project, fun to play around with and fun to work on, right? So that's smoke simulation. Okay. Another one, so this is sort of an add-on to rendering. This is some real-time ray tracing. So I'm gonna let this video play, but essentially we're sort of going through this scene and rendering all of this in real time. And you can see some of these, ray tracing artifacts. You can see sort of light bouncing through this cube onto the wall, reflecting around all the different reflections. It's sort of really cool and running really, really fast. So a very interesting project. If you have GPUs or something you want to work with, this could definitely be something you think about. Okay. Let's real-time ray tracing. Okay, and then another one is sort of non-realistic watercolor shading. So this is sort of a different type of, rendering, texture mapping sort of thing, where you're not necessarily trying to, up until now, most of our rendering has been sort of photorealistic. We want to get as realistic as possible. This is sort of a different approach where it says, we're just trying to get it in some sort of aesthetic. So it could be watercolor. You could have make it look like a comic book. There's all sorts of different things you could do that could be really interesting and could be kind of fun to work on. So I think that's it. Yeah. So that's all the examples I have here. Once again, like I've said, not at all exhaustive. So definitely feel feel free to sort of think beyond what I've mentioned here. And yeah. Okay. Oh, I see there's some questions in chat. Yeah, I'll put the slides on Piazza afterwards. And yeah, cool. Any other questions on this? And we'll put more like of the logistical details on Piazza right after lecture. So, you can check there for the details about like the deadlines and groups and stuff like that. What was my project? Oh, my project, when I took this class, I did like a melting simulation. So sort of like having different metals and then like applying heat and then they would slowly melt. So yeah, it was kind of interesting. Cool. All right. If there's no other questions, let's pivot real quick. I'm going to stop sharing that and start sharing this instead. Okay. So the second thing we want to do is go over the results for the first art competition. Thank you very much to everyone who submitted. It was really fun seeing it, seeing what you all kind of came up with, what you all worked on. It was very cool. And then thank you also to everyone who voted. That helps kind of keep this competition going, makes it interesting. So yeah, thank you to everyone. And without further ado, here are the results. So first off, whoops. All right, yeah, in third place, Parth with his jumping jack cube man. So Parth didn't feel like exercising, so cube man did it for him. Cool little gif made with a couple different SVG files. So really great work, Parth. Looks nice. All right. And then in second place, Buff Kirby by Claire and Fire. So really, really fun to look at image. They took a PNG, sort of turned it into triangles, and then rendered it. Really cool and really nice work. Okay, and then last but definitely not least, first place winner. So Yimo for the beheading of Cube Man. So revolution here with two robots.svgs drawing. Really cool all around. And whoops. Yeah, okay, I guess that's it. But thank you all for submitting these. They all were really fun to look at. And yeah, definitely keep participating in future ones. All right.
Super. Thanks, Rishi. Yeah, thanks. Great examples, great slides, Rishi. Just to maybe verbalize my answer to some of the questions about the final project. So let me generalize the answer that I gave. The question I got from Dhruv was specifically about like games and using game engines, which could simplify a lot some of the implementations of what you saw in some of the game demos that Rishi showed. Here's the deal in the final project. We don't have rules in the final project that say you have to start from scratch or build the entire thing from, like a blank piece of source code. You can choose whatever starting point that you want. Now, so if you look at it one way, you can be like, oh, then I could build a game really easily. It'll only take me 5 minutes and then I'll be done with my final project. And that approach, you wouldn't take very much time. Your team wouldn't learn very much and you wouldn't get a very good grade in your final project. So that's would be the wrong way to look at it. The right way to look at it is you can ballpark the amount of time that it would make sense to spend on this project by being proportional to the size of the team and the duration of time. Compared to, for example, your assignments. So your assignments are pairs for 2 weeks. This is the final project is 4 people for 4 weeks. So you can kind of get a sense of the kind of scale of effort and team bandwidth that you have at your disposal. Then wherever you pick your starting point, if you pick it starting on a game engine that already exists, for example, or if you say, hey, we decided to start from scratch and build a game as an example from like a blank source code page as our starting point, wherever you start from, you have 4 weeks from which to build to your endpoint and to articulate what your goals are for learning and for achievement. So it's critical for you, and you will learn different things depending on whether you start from scratch or if you start building on top of something like a significant library or platform that already exists. Okay, so that kind of makes sense to you. But in this project, in this class, you have freedom to make that decision. What's really critical though, and many teams don't always do this well enough, so let me sort of try to make this clear here, and we'll try to reiterate this as we go through. It's really critical for you, given that sort of an open freedom, that wide open platform to choose your starting point. It's really critical in your presentation, like your proposal and then your final presentation and video, that you make absolutely crystal clear where you started from and then exactly what your team built on top of that. Does that make sense? If you don't do that, then like you said, somebody look at your final project and be like, oh, I could have built that in 5 minutes on top of this existing game engine that I know about. Okay, so that's just basics and sort of communicating the starting point and the end point and what your specific contribution was for your team. So this is some high level sort of comments. Okay, all right. We can talk more about it, about your specific project ideas and any questions you may have about those, about that setup and format as we go along. Okay. All right, why don't we continue with color? Let me just share my screen here. Okie dokie. All right. So last time we had finished this introduction about color, all of these sort of weird perceptual characteristics of color, some illusions, if you hadn't seen them before, that are pretty striking. Perhaps, hopefully at least one of those for each of you that you hadn't seen before that was surprising to you. A lot of those really bowled me over the first time that I saw them, to sort of give you a sense on how malleable our visual perception is, how fine-grained it is, how many different sort of aspects of it that are going on at the same time, how complex and in many ways how weird it is. And yet at the same time, as I finished observing towards the end of the last lecture, in engineering for color, we really know, right, how to engineer color reproduction so that on all the displays that we're looking at, the photographs that we take and show, or the documents that we create, like the presentation I'm showing you today, and that we broadcast through displays to other people, the color reproduction on it, we know from our experiences with these kind of devices, is really convincing. The color looks the way that it does in reality. If we look at it together and talk about it, yeah, this looks red, this looks orange. If we compare it across displays or to the real world, it has the fidelity that we find very convincing. So how does that color reproduction work is the thing that we started to talk about. And we start to lay some of the foundations for understanding that in terms of the physical basis for color, a model for light detection, right? And then I started to talk about tri-stimulus theory. So we finished like a single, a model for a single detector of light that aggregates all the light that arrives there. With different sensitivity to different wavelengths. And then I said, hey, in terms of tri-stimulus theory of light, theory of color, the difference between having a detector that points at the world and sort of adds up light with a single detector versus tri-stimulus, or the 3D nature of color, is that in our eyes, we have 3 types of these detectors. We have 3 types of photoreceptors. So I'm going to talk about the biological basis for that in just a few minutes. But before the folks figured out all of the details of the biology, a lot of this was characterized experimentally in attempts to empirically measure or lay down empirical systems for measuring color. Okay, so a lot of that came from Maxwell, as I described in the famous color matching experiment, the fundamental color matching experiment. I described this spinning top experiment last time. As a reminder, what Maxwell did was to run a series of experiments where you spin a disk like that's on an axle here, and then you get sort of 2 regions where colors are going to blur. In this outer donut area that you can see here that has red, green, and blue sectors, those are going to blend together and form some kind of average color for that proportion of red, green, and blue. And if you sort of look at this, you can visualize that you can sort of slide those pieces of paper sectorally so that you can end up with different fractions as you want. And then in the inner pie here, in this case, it's black and white pieces of paper. They're going to blur together, and you can tell it's going to make a gray color. So this is an experimental setup that's designed to enable someone to find proportions of color, in this case, red, green, and blue papers. Of course, you could put whatever you wanted on the outside, that when averaged together through the linear summation of light, the attempt is to make it match an inside test color. Okay, so there's a test color on the inside, there's a matching color set on the outside, and you adjust the proportions of the colors on the outside until you match. The key aspect of this that was in many ways not obvious, but is really at the central part of the nature of measuring color in a rigorous way. The key idea is that we're going to do it by a perceptual equality of two colors. That's the bedrock principle under which we're going to build a whole colorimetric or measurement system for color. That was Maxwell's key, the fundamental scientific and engineering insight. Okay. And it turns out that for under appropriate conditions that I'm going to qualify with sort of the important boundary conditions, basically, if you have three colors that like red, green, blue, you can span basically any test color in the middle. Okay. Subject to some boundary conditions related to gamut that I'm going to talk about. Okay. And that finding is very important. If you have only two colors, you cannot do that. You can't accomplish that matching to any test color in the middle. If you have four, you can do it. You don't need four. It turns out that three is the magic number. Okay. Okay. So this was a very early version of this color matching experiment. This has been modernized in various ways. I'm going to show you a more modern version here. I'm going to get into these details in a second, but let me just check the chat. How do we know what pure RGB looks like, the colors that excite the three cones the most? Or does Maxwell approximate them here? Yeah, good question. So RGB is chosen here, but it turns out there's quite a bit of latitude in choosing what those three colors are, especially once we get into a linear systems theory for this that encompasses the ability to handle colors that are outside the gamut. Important technical detail I'm going to go through in detail in a bit. But one point to make clear here that I need to make clear is that red, green, blue are not the colors that most excite the three cone cells in particular. Okay, that's not true. We're going to go through the biological basis for that in a little bit, and that may become clear. But even what red, green, and blue are is, as we think about the physical basis for light, the spectral power distribution that is emitted from this red, green, and blue pieces of paper, even that is, as we talked about, going to depend on the nature of the illumination that's falling on it. So if it's in bright sunlight, recall that we have a certain spectral power distribution that was trending higher towards longer wavelengths. That's going to reflect off these red, green, and blue absorbing surfaces, spectrally absorbing surfaces, and give a different emitted spectral power distribution compared to if we looked at them under, for example, like a blue sky in the shade of a building. We would still call them as a person red, green, and blue. Okay, we wouldn't be confused about that because of the chromatic adaptation nature of our human visual system, as I described last time. So the pieces of paper would still look red, green, and blue, but the spectral power distributions of them would be completely different. Nevertheless, we can still talk about whether they would match the appearance of the inside test color under this spinning superposition color matching setup. Okay. Let me show an example to sort of continue talking about that in more detail. This is a color matching experiment setup that I want to describe to you in detail. It's similar to the setup before in the spinning wheels, but it's a little bit more formalized. Okay, it's a little bit more controlled. So here's a human subject looking into a color matching experimental setup that you can see here. He's sort of looking into a little room. The room inside is dark except for a few light sources. As you can see, there's a test light that is shining here onto this part of this room. And there are three what we call primary lights. These are the lights that we're going to adjust the brightnesses of in order to try to match the test light. In this case, again, it's red, green, blue, but that's not a necessary condition. They could be whatever color, whatever spectral power distribution light sources that one wants to have for primary lights here. These shine onto this portion of this color matching room setup. And again, the subject is going to have controls to be able to change the intensity level, the brightness of those three primary lights. the person looks into the room through a hole. It's also sort of important. If you imagine looking in through a hole into this room, what are you going to see? This picture here shows it. So the subject looking inside, this is sort of the hole in the wall, is going to see on the left half the test light, and on the right side, the superposition of the three primary lights. Okay, this is called a bipartite field. Bipartite just means partitioned into two. Okay, so a left half and a right half for the test light and the primary light. And it's set up in this way so the subject sees the two of them in close proximity and can do a clear visual judgment about whether they're exactly the same or not, and adjust the primary light the primary light ratios until the subject sees the appearance of them as identical. In addition, since they're looking through a hole, you have to sort of consider what they're seeing in the surrounding. Okay, so if you put like a big colored poster on there, like that was very brightly colored, and you see a brightly colored poster and you look through this and see some other colors, maybe they might be a bit sort of confusing color-wise. So usually it's standardized to make like a you can see in sort of this black part here called the surround field. It's say a piece of paper that's white and then that's illuminated by a light source that's a good approximation for white. Those are all standardized too. A very common standard for white light is called D65. That's a good approximation for the spectral power distribution of daylight. Okay, right. And so Ray asked that question exactly. What is this light source? So it's just something to shine onto the surround field. It doesn't in any way change the color because of the setup of this and the way that the light is blocked. It doesn't in any way change the color of the test light or the primary lights. It's just to set sort of a white donut through which one can look at the rest of this. If you think about this in the context of chromatic adaptation, the other way to sort of think about it is that it's setting up sort of like a viewing environment where the viewer, the subject, gets to have a sense of, okay, this is under normal lighting, okay, like normal white light, if you will. And so the test colors are in that sort of isolated setup. You could also, of course, do studies where you do the surround field in crazy colors for chromatic adaptation studies. That's, something like maybe Professor Mark Fairchild might do some research on. That's not what we're talking about here today. So the standard condition is with a white surround field D65 light bulb. And that's the situation that we'll use too. Okay, so this is the color matching setup. Let me walk you through an example of how the color matching experiment might go for a subject. Okay, so let's say they look into that field. Here's redrawn just as a bipartite field that's square, but in the real setup, it's usually circular, but the principle is the same. So on the left-hand side, they see a test light, and this might look sort of light blue or light cyan to you. On the right-hand side, all three primary lights off at first. Okay, so the subject gets to twiddle the dials on the powers of the three primary lights, P1, P2, P3. Notice here, they're not written RGB anymore. They're just three primary light sources. And in fact, the subject may not have any idea what the primary lights specifically are, except by observation, by turning the lights up, they could get a peek into it, but they don't necessarily, they don't know what the spectral power distribution or is anything else. This is purely a perceptual observation experiment procedure. So the subject gets to twiddle the dial, says, okay, this is getting a bit closer, but it's kind of dark. I'm going to try to twiddle it a bit more. Okay, that's kind of getting closer. Maybe they're a similar sort of color. They're both teal or something, but it's not quite right. So they sort of tweak it some more and they say, okay, I've tweaked it now, and I've got a primary color ratio, P1, P2, P3, for the intensities of those three colored lights that gives me an exact match with the test light on the left-hand side. This is completion of this one trial of a color matching experiment. Okay, so they say that they're done, and we write down for that test light, P1, P2, P3 is the match. Okay? And you can imagine doing this for any test light that you want to do here. And I'll show later there's a way to sort of systematically do this for the entire spectrum, sort of wavelength by wavelength. And that gives a sort of exhaustive measurement of any possible wavelength from which we can use math later to calculate what the matching ratios for these test lights would be for any possible color light that we might put in. I'll come back to that in a couple of slides. Okay. Someone says, I don't think this test can give exact numbers. Yeah, that's true. It's a experimental procedure. So, like all experimental procedures, it's going to be testing variation. There's going to be a real world variation in sort of the judgments that people have. But let me say that the discrimination of when you get an exact match is a very fine perceptual discrimination task that is very repeatable for most people. There's, of course, error bars perceptually. It's not infinitely accurate. but this is something that from person to person individually, for a person individually through repeated tests. And then amazingly, across people, the color matching results are surprisingly robust. It's actually at the basis for the color reproduction accuracy that we all enjoy in the modern devices that we use today. Okay, let me show you one thing that happens though. What I showed you before here can happen for lots and lots of light, the test colored lights, and can work great. Here's something that always happens though, that is important to know about. And I want to walk you through what happens and then talk about the mathematical implications of it. What's happening here is that the test light is out of gamut. So we can't actually come up with a match. Okay, so let's see what happens. So this is the test light. The subject starts to twiddle the dials, get something that's approaching a similar color, maybe a dark pink. Twiddles it some more and says, okay, now it's getting red. It's kind of similar. But then experimentally, what the subject finds is they can't adjust the dials anymore to get any closer perceptually. Okay, so that's what happens. So they might try to, increase P2, try to turn down P1, try to turn down P3, turn them up, and they just sort of go through all those combinations and come to the clear conclusion that nothing is helping. Like this is as close as they can make the two colors, and they don't match. Okay, as you can see, this will happen. And when this happens, the technical description of it is that the test light is out of the gamut of the three primary lights. What happens in this situation is that it turns out in order to create a match, I'll describe what happens. It might seem a bit strange at first, but then we'll go through a mathematical explanation of it, and perhaps that will, make more sense and seem more theoretically satisfying. So what happens here, to resolve this and achieve a match and get numbers that we can write down, what happens is that the subject is allowed to and finds that they have to move some of the primary lights onto the test light side. Can you see what happened here? The subject took P2, the second primary light, and added it onto the test light side, and then adjusted the intensity of P2 until a match was achieved. So P2 is completely off on the right-hand side, and it's now turned on the left-hand side, which is strange, right? That didn't happen at all earlier. So it's as if we've taken the light and we've added it onto the test light side. It means that the test light is a different color now. Okay, so that's empirically what happens in this experiment when the test light is out of gamut. And one way to think about mathematically what's happening there is shown up here. You have a positive amount of P1 according to the amount here, and P3 as according to the amount here. But the amount of P2 is actually negative. So moving it onto the other side is mathematically equivalent to having a negative amount of light here. You could, if you want to think purely abstractly, you could imagine it as if you had just the test light here and you could somehow deliver negative P2 light, then you might create something on the right-hand side that matches. But of course, physically, there's no way to do that, right? We can't deliver negative light into the scene. Does that make sense? So mathematically, this is what's going on, but the only way to actually have it appear physically, empirically in the experiment is to do it as I described. Okay, but actually, this mathematical interpretation from an abstract perspective is completely valid and is very useful to think about because it provides a complete linear systems view into what's happening from a color perspective. Okay, it's exactly right. Dhruv says it's sort of like finding a linear combination of basis vectors. And if you have a linear combination, if you have basis vectors that span a linear space, you have to be able to provide linear combinations of them for all real numbers, not just positive or not just for non-negative numbers, but for all real numbers, including negative numbers too. So this color matching experiment, the only way to express those negative contributions of light is by the method that I showed there. Okay, that negative color matching, as it's called in terms of the experimental procedure. Now, what one finds in the color matching experiment, this is one of the most important characteristics of it, and it's a little different than I described in previous slides in the lecture where I talked about the physical basis of light and then measurement with devices and superposition, for example, in a monochromator or in a spectral radiometer for superposition of light. This one is the linearity of the color matching experiment, which means that it's perceptually adding up linearly. One finds that to a very high degree of accuracy, the perceptual color matching experiment is also linear. It also obeys superposition. So, for example, if one has a subject and the subject makes a match of teal to this ratio of primary lights, for example, and then you give the subject this pink and they form this color matching ratio, then by superposition, if you just add these two colors together, it won't look, like half cyan, half pink. It'll be the linear combination of teal and pink, whatever that turns out to be in the test light side. And you just get the subject to do color matching from scratch, you will find that they form the ratio exactly as shown here, exactly as superposition of these ratios that they found individually. Okay, so that's a pretty remarkable fact, and it's saying something about the perceptual superposition of our color perception. Okay, which is not necessarily obvious from the biophysics background that we're going to go through in a little bit. Okay, so those are some of the key facts about the color matching experiment. I want to take one slide and talk about what is the dimensionality of human color perception, at least as we can deduce from this color matching experiment. Let me pause for 1 second. Just take a quick look at the chat. Okay, sorry, let me go back for 1 second before getting to this slide about dimensionality. Stravia asks, so based on what I described earlier, exactly what P1, P2, and P3 are don't matter too much. Yeah, that's the right big idea, okay? You could pick almost any three colors, taking into account this negative color matching requirement and thinking of it as sort of adding linear combinations that may include negative portions. And conclude that, yeah, almost three colors would work. Now, of course, you could pick, two greens that are almost exactly the same and one red, and then you would have some numerical problems. You can think of it in linear systems theory as having two vectors that are very close numerically to being linearly dependent or something, basically the same. Or you could have two vectors and then a third one that is very close to like a linear combination of the first one, and that would also cause you a problem. So you need some linear independence. You need to accomplish linear independence. But in this very high dimensional space of 1D spectral power distribution functions, it turns out that picking almost any three colors at random in terms of spectral power distribution will satisfy linearity. Okay, so that's a pretty powerful and important result. What does the light pink, blue and pink on the right mean? I think I described it. Yeah. So this just means the adding together these two colored lights in superposition according to these ratios. Okay, great. Let me go on. Okay, so what is the dimensionality of human color perception, and how do we know? I think that's a really critical question, a deep theoretical question that you might want to ask yourselves. We kind of quote unquote know that if you have quote unquote normal color vision, that it's 3D. We all know that displays are RGB and all these things, but if you sort of leave that on the side, how do you know? How do we know that scientifically? What's the basis, the theoretical basis for that belief? Okay, in fact, along those lines, like, what's the definition of dimension, of color perception here? We sort of know it's 3D color perception, but like, what does dimension even mean? And we've already been talking about this, so it's easy to continue this. We can appeal, and I encourage you to think of it in this way, we can appeal to a linear systems theory where the colors form a linear space through combination, and we're going to accomplish, well, let me be a little precise about that. The colors are the percepts that a person sees, right? So that forms a linear space that we can span by having three representatives in the color space and then taking linear combinations of them. And so the question of dimension will be equal to the rank of a basis for that linear space. What does that mean? Because there's a few mathematical technical terms in there, right? So what's the rank? The rank is just the number of vectors, okay, in a basis. What's a basis? A basis is the minimum, is a set of vectors that spans everything in the space. It has no linear, and they're all linearly independent, okay? So this reduces our search here to how many, what's the basis for this linear space? This is an empirical question, and it can be answered with the color matching experiment through a series of a battery of tests. We didn't do the battery of tests here. I just showed you a few examples. Let me describe to you some of the key results if one does this color matching experiment and does it for a whole series of test lights and primary lights. In the color matching experiment, one empirically finds that for subjects with quote unquote normal color vision, so the bulk of the human population, 3 primary colors are necessary and sufficient to match any test color using the procedures that I've described to you already. You can have 4 primaries. Okay, so I drew with 3, but that's sort of a variable. You could have 4 primary lights there, 5 primary lights there, 2 primary lights there. 4 or more will work. You can find a ratio of them that will match any test light, but it's not necessary. You can do with fewer. And 2 primary lights, crucially, 2 don't work. You can for sure find a test light with only 2 primary lights on the other side that you can never match that test light. Okay. So this is a way, if you read between the lines, of saying that 3 is the rank of the basis that's required to span this linear space. Interestingly, for, say, red-green colorblind subjects, okay, and we talked a bit about that last time, you find something different. Only 2 primary colors are necessary and sufficient to match any test color. 3 will work, but is not necessary. So in this way, we can find that the dimensionality, color dimensionality for perception for a colorblind person, at least for red-green colorblindness, is 2. Okay. A question that I got here. So here we allow negative coefficients to the 3 primary colors. Correct. Yeah, that's exactly correct. Okay, so in the color matching experiment, the way that the negative comes up is, as I described, you have to put it on the other side in order to complete that experiment test physically each time. But the interpretation of it is that means we have a negative contribution of that primary light, and that forms the linear, the linear basis for superposition of all of these lights in a linear system sense. Okay. Let me just finish up by describing how you can exhaustively do this color matching experiment. I alluded to this before. Let me do it in the context of actually just telling you about one standardization procedure for all of this. So as I described, you can choose any 3 primary lights that you want. And when you think about that, that's a very big choice, because even though I showed it before as red, green, blue is not a full definition. Remember, the definition of a color source in a physical basis sense is the spectral power distribution, right? It could be the blue sky that you sort of, like bounced off a mirror into the system, or something that's coming off like a tungsten bulb for yellow or something like that, okay? So exactly what is the spectral power distribution is important to specify if you want to fully define the primary lights for a color matching experiment. The CIE standardized this in the so-called RGB standard by defining a red, green, and blue standard color source for this standardization procedure, okay? So they defined 700 nanometer, 546.1 nanometer, and 435.8 nanometers, which are sort of convenient sources relative to emission, like laser emission sources that are at those wavelengths, to define a red, a green, and a blue primary light. So these are the 3 primary lights used in a color matching experiment. The color matching experiment is done exhaustively, wavelength by wavelength. So the test light is also a monochromatic light, but it's one where you can sweep it over any wavelength systematically, one at a time, okay? So you start at short wavelengths, 400 nanometer, then go to 405 nanometer, 410 nanometer, and so on and so forth. Sampling every wavelength of the visible spectrum. And for each one of those wavelengths, you do a color matching experiment. You get a human subject to find the ratio of these 3 color primaries to match that monochromatic test light, okay? If you do that, what's the data going to look like? It looks like this, okay? You can graph it. The graph is on the horizontal axis. It's the test wavelength. Okay, so at 400, that just means I put the test wavelength in at 400 nanometer, and then I asked the human subject, to do the color matching experiment. And what did they do? They matched it to basically almost no red and green, and a certain amount of blue, and so on and so forth across all of these. You can see that for certain wavelengths, for example, 500 nanometer, the matching involved a negative amount of the 700 nanometer. So you know exactly what happened there. Okay, it was out of gamut, and the negative color matching procedure that I described earlier was needed to be applied in order to find a full color match. These data, as they're presented here, is just 3 graphs, right? These are called the color matching functions for these 3 primary lights as a set. for every monochromatic light source forms a complete description for let me make sure I get the description exactly right. It fully characterizes any possible spectral power distribution of light with respect to what the matching values would be for these specific primary lights. How do you get that? Because this is sort of wavelength by wavelength. If I gave you a particular spectral power distribution, okay, one that wasn't in the test set, okay, you give a graph of the spectral power distribution, you can just form, you can sort of figure what the red, green, and blue matching ratios would be by taking superposition. So wavelength by wavelength, you can say what is the proportion of that RGB weighted by the amount of the spectral power distribution, and you sum them all up. Okay, you take those integral projections. And by doing that, you can calculate, you don't have to put that specific spectral power distribution in front of a human subject and get them to do the matching experiment from scratch. You can calculate by linear superposition what their match would have been from this one experiment. So this is a way of sort of, sometimes it's called gauging the spectrum. It's a way to exhaustively measure what the color matching would be for any sort of set of primary lights. I got a question here. Gregoria asks, what do you mean by 4 primary color lights when there are only 3 primary colors? Yeah, so thanks for asking that question. Let me just go back here. So what I mean by 4 primary lights is that in this color matching experiment here, let me go back here. It's drawn here with 3 primary lights, but 3 is not necessary. You don't have to put 3 there. You could put 4, you could put 5. That just means that you have 4 or 5 dials where you control the intensity of those light sources. The game is exactly the same. The color matching game is exactly the same. We provide a test light on one side, and we ask the human subject to adjust the intensity of the set of primary lights, whether it's 3, 4, or 5, changing the intensity of them until the human subject says that these 2 colors here, the primary light ratio or combination and the test light look exactly the same. Okay, so the primary lights do not have to be 3 in the color matching experiment. Thanks for asking that question. Yeah, I realize these slides usually show with 3, and that can be a bit confusing. Okay, all right, so that's color matching. Let's switch gears a little bit now and bring in the next dimension of knowledge about this, which has to do with the biological basis of color. Okay, so now we're going to go into the eye and look at some detailed specifics, specifically like the clone cells that we've touched on a little bit and some of the photopigments that are involved in our human sensing of this spectral power distribution of light and how those contribute to our understanding of color. Here's a cross-section of a human eye. So the key thing to note is it's kind of like a camera optically in a lot of the ways that we've studied in cameras. So this is a lens system on the left-hand side. There's the cornea that is a curved, the curved outer part of our eye that's visible and touching the air. Inside, there's the thing called the lens. In this case, it's a anatomical body that is kind of squishy so that it can be pulled by these muscles that you can see on the sides. And if it's pulled, then it gets flatter, less curved. And if it's allowed to relax, it will bulge out more. And that's how we can change the optical power of this optical system here and allow us to focus close or far in the world. The net of this, though, you can think of very much like the camera lens systems that we looked at when we were looking at cameras and lenses. The light passes through this pupil and is brought to focus as an image onto the imaging plane, which is at the back of the eye. A few things that I'll highlight. I won't go through all of the specific anatomical parts, but just pick out a couple. There's the retina, which is, of course, the part of the eye that's photosensitive and that is going to be receiving this electromagnetic radiation through the pupil and then converting it through a process that we'll talk about into signals that eventually go to our brain. The most dense part, cellularly speaking, of the retina is called the fovea, or the fovea centralis being the absolute highest point. That's the part that has the most color-related cells packed the most tightly. And that's where we will position our eyes, optically speaking, right? You can imagine that chief ray being drawn from the fovea centralis out the pupil, and we will move our heads to position that ray or turn our eyes in their sockets in order to put it where we need to see the most detail. So as we read, we will move our eyes in order to position the fovea centralis on each word and letter scanning across as we read. For sort of trivia's sake, there's a blind spot here. Okay, you probably heard of this before. Maybe you've done some, looked in a book and seen some things that help you close one eye and you can actually perceive your own blind spot. The reason there is this blind spot is that on the retina, after the absorption of all of this light, there's some processing through like layers of neurons that eventually have axons that all congregate and go out the optic nerve that go to the brain. So that's how all of this visual information is eventually passed through to our minds. And as they exit, it just turns out if you draw the wiring diagram, there needs to be a place where they all exit, all those wires, neuronal wires, axons, where you can't position any photoreceptors because it's too crowded in the wiring diagram. Okay, so that's the nature of the optic disc and the blind spot. For our purposes, going back to color, I'm going to talk about the retina now and cells on the retina. So the key point is that there are photoreceptors on the retina, and these are the cells that do the key work in absorbing the light and then making it available through to the neurons that I described that eventually will encode the information and send it to the brain. There's two types of photoreceptors, or two classes of photoreceptors. They're called rods and cones. You may have heard about this before. The reason that they're called that is that if you look at it under a microscope, the rod cells look kind of like cylinders, and the cone cells kind of taper towards the end. Some of these are a little banged up, I think, probably in the procedure that harvested these cells to put them in an electron microscope. But you can see this one, for example, is kind of tapered to the top. This one a little bit too. These are cone cells. Okay, so it's just because of the anatomical shape of those. But functionally, they're very different. And I want to describe the key aspects of those. So the rod cells are active basically on only very dim light conditions. Those are technically called scotopic conditions. You can think of that as like dim moonlight or darker. So very dark conditions. Okay. There's a lot of rod cells, like 120 million or so per eye. And in those dark kind of conditions, rod cells give us a view of sort of the intensity of the world. We can see edges, we can navigate, to move around, but we perceive only shades of gray. We don't have any color sense in those kind of conditions when only the rod cells are active. The cone cells are the primary photoreceptors in typical light levels. Those are technically called photopic light levels. So wherever you're sitting right now looking at your display, you're in photopic light levels, okay? Remember, it's only when you get to very dim moonlight that we get down to scotopic light level conditions. Everything above that will be in photopic light levels, and that's where cones are active. There's far fewer cone cells per each eye, 6 to 7 million cone cells per eye or so. The key thing here is that there are 3 types of cone cells, and each of them have a different spectral sensitivity that I'm going to go through in detail, okay? So there are 3 different cones that have 3 photopigments in them that are slightly but crucially different, okay, photopigments. Those come from 3 different separate genes in our DNA. Okay, so this is real fundamental stuff. The reason there are 3 comes from our DNA, okay, and it's encoded in our genetic material. The 3 types of cone cells are crucial in providing our sensation of color. And as was asked earlier in the chat, yeah, this is sort of the biological basis for the 3-dimensionality, okay, of our color. Although that was not a critical thing, that was not a trivial thing to establish scientifically in the history of this science. Let me, okay, go on here. So this is a bit more trivia, not really critical for where we're going, but it's useful to know a bit about the eye and landscapes and just mapping where we are in it. This is the fovea centralis. I said it's the place where the cone cells are the most dense. So you can kind of get a sense of that density here and the size of the cone cells, which are relatively small. This is at, say, 0 degrees eccentricity. Eccentricity just means distance on the retina from the fovea. In this case, it's plotted as a function in millimeters. So approximately, I don't know, it looks like 1 millimeter away on the retina is quite different. The cone cells are a lot larger. And as you can see, this is a graph of density. So it's very, very dense, the cone cells in the fovea, 150,000 cells per square millimeter. It falls off very rapidly, as you can see in this graph. So a millimeter away, it's down to like, 15,000 cone cells, like maybe a tenth. You can see kind of the shape of the cone cells here. And as you get further away, in this case, like, I don't know, 6 millimeters away from the fovea centralis on the retina, the cone cells are even further spaced apart, and you can see more rod cells. Okay, so this is a rough sort of landscape. So the key thing to know is that in your fovea centralis, you have a lot of really finely, densely packed cones. And in your periphery, one of the reasons that you can't perceive as much detail in your periphery or your peripheral vision is because the cone cells that are active in photopic light conditions are just not very dense there. Another key thing is that there's no rod cells in the fovea, which is important. And so in the place where it's most important for photopic regular light viewing, we have only cone cells and we have color vision there. Okay, let's talk about these three types of cone cells. This is really the critical stuff with regards to color perception, okay? On the retina, there are three types of cone cells. They're colored in this diagram, red, green, and blue, sort of for color visualization, sort of, convenience, so you can see spatially how they're distributed. Do not confuse this with the three cone cells primarily, receiving red, green, or blue light. That is not what's going on. I'll talk more about that. Okay, the three types of cone cells are called the S, the M, and the L cells. The reason they're called that corresponds to the fact that they have a peak spectral response at short wavelengths, medium wavelengths, and long wavelengths, okay, S, M, and L. What does that mean? Let's take a look at their spectral sensitivities. So they're shown here. This is the response curve for the S cone cell as a function of lambda, the lambda being the wavelength, okay? So you can see it has a sort of bell curve shaped. It has a peak in short wavelengths around, under 450 nanometers. This is the M cone cell, or the middle wavelength cone cell, this green one here. It has a peak around, I don't know, I've forgotten what it is. I think it's in the neighborhood of 540 nanometers or so. This is the L cone cell, as you can see here. It peaks slightly higher, probably around 560, 570 nanometer or so, okay? What you observe is that each of these cone cells absorbs light over a range of wavelengths. It has a peak, but it absorbs to fractional amounts over a pretty wide distribution. Recall that this is very similar to the model individually. Each one of these is very similar to the simple model of a light detector that I described in detail last lecture, okay? The other thing that you'll notice is that these three spectra are not separated, okay? It's not like S on one side and then, completely separately, there's an M, and then separately there's an L. If you look at these, they completely overlap, right? And in fact, if you look at the M, it sort of seems almost degenerate. It's like very closely tied and almost like completely overlapped with this L cone cell. These are all facts, okay? This is the nature of the spectral distribution. Again, this is true across a very broad section of the human population, and it comes from genes that have actually been sequenced and have been exhaustively measured in their spectral response characteristics at this stage after decades of scientific work. So these are encoded for in our DNA, okay? So instead of one detector, one simple detector, as I described last lecture, on our retinas, we have three types of photodetectors, and they have slightly different spectral responses, as you can, or quite different spectral responses, as you can see here. We can talk about the scalar response of each one. Each one is a simple detector, okay? We can talk about the capital S, capital M, capital L response, which is a scalar number. And it's exactly the model for the simple detector that I had last time, where the response of it, remember the response was the probability that at a specific wavelength that detector would detect a photon. R sub S is exactly that sensitivity curve for the short wavelength cone cell. Similarly for R sub M, that's the sensitivity curve for M cone cells, and R sub L over here, okay? All right, so those are the fundamentals of the detection spectral characteristics. Let me show you an example of what this would look like, okay, for an image. Okay, so let's say you look out in the world and you see a damselfly. The damselfly is being optically focused onto your retina. So what happens? Let's say it's projected onto this portion of retina, okay, for argument's sake. So here's a collection of cells. Here's a portion of that damselfly. So it's the wing portion and part of the body. It gets projected onto this portion of the retina. So what happens? We go cell by cell. This is biophysically what's happening. Let's pick this cell over here for argument's sake. As you can see, it's red, so this turns out to be an L cone cell. So we get this response as the activation level for that. Think of it as a single detector, scalar detector. That particular cone cell is going to be the dot product or the integral projection between two things. The first is the spectral power distribution at that XY location. Okay, so that's a function of wavelength at this XY location. Recall that it was a beige background behind this damselfly. So this spectral power distribution is, I guess, representative of that beige light. And then the response of this particular L cone cell is the R sub L function we looked at before. So we just take a dot product between those two vectors or this integral equation, equivalent mathematically. This M cell over here, which is also in this beige light regime, okay, has a similar but different response to the M cone cell. The difference is that it's being projected against this M cone spectral response, okay, which is different than the L slightly. This is an S cone cell over here, and you can tell that it's going to have a quite different response because the S cone response is much shallower and is also at shorter wavelengths. Okay, so cell by cell, every cell, we have a spectral power distribution, we have a response sensitivity as a function of the spectrum, but the output of the cell information-wise is a scalar number, okay, for that moment when you're looking at this moment where you're looking at this damselfly, and this is happening at every cell over your retina. So, and again, so this is the mathematics for each of those three, for those three cells. And I said you could do it as dot products. Let me just remind you of that math because we'll use some of this later when we talk about a color reproduction in a matrix solution sense. So S is this integrals over here. If we write R sub S and S, these 1D functions as vectors, then we could just take dot products between them. If we write them as column vectors, column and row vectors, then S is a tall column vector, right, representing a sampling of the S spectral power distribution at every wavelength. And R sub S will become become a row vector. And if we write SML as an output 3x1 vector for the output of the eye at any particular location for a given input spectral power distribution, then we can write R sub S, R sub M, R sub L as stacked row vectors here, and the matrix math will work out to give us the SML responses exactly the same as these dot products over here. So this is just a different way to write the mathematics that we'll use a little bit later. Okay, so this is matrix formulation for that. Let me back up for a second and just let's reobserve what's going on at a high level here, a conceptual level, an intuitive level. This is really crucial. We have a dimensionality reduction occurring here from infinity down to 3. Last time, last lecture, I said if you have a simple model for a light detector, it was from infinity down to 1 because that detector could only put out one scalar value for whatever spectral power distribution was falling on it. In this case, when the light falls onto some neighborhood of your retina, there's a lot of cells there because they're very tiny, and some are S, some are M, some are L. In that neighborhood, that spectral power distribution that arrives there is going to create 3 measurements, 3 scalar measurements, 1 S, 1 M, 1 L, and those 3 will go to the brain. But recognize again that we don't get almost any information about the original spectral power distribution. We only get these 3 scalar projections of it. So this is an enormous information reduction that's occurring here, okay? Now, so this is from infinity to 3, infinite dimensional to 3-dimensional. That's in the spectral sense. Just to be really clear about this to avoid confusion. What about, these are 2D images, right? We looked at a damselfly that's 2D. So what is this 3D infinity or versus 2D? The infinity to 3 is in terms of the spectral, in terms of the wavelength function or the spectral characteristics. In terms of the 2D image, yes, it is a 2D image of the damselfly. The dimensionality reduction that I just described is occurring at every 2D position in the visual field, okay, of this picture. Okay, I'll continue this. Hopefully I won't beat this horse too much to death, but just to reiterate again, the human eye does not measure and the brain does not receive information about each wavelength of light. Rather, the eye measures 3 response values only, 3 scalar values SML at each position in the visual field, and that is the only spectral information that is going to transport to the brain, that'll be available to the brain. And that's the result mathematically of integrating the incoming spectrum against the three response functions of the S, M, and L cone cells. Okay, so we have a spectrum, could be a very complicated function, goes into the eye at any one particular location where that spectrum appears, it projects onto SML, and the brain for that spectral power distribution receives three numbers only. And that's the information from which it will make a 3D, or it will make a color judgment. You should expect from that description of the world, that huge data reduction, that a lot of different spectral power distributions might look the same. That'll be the right intuition. That's exactly the right intuition. Many, many, many different spectral power distributions will look exactly the same to the brain because the brain is going to receive the exact same three SML numbers for them. That phenomenon is called metamerism. It's a very important concept, crucial to practical color reproduction in all our engineering systems of today. Let's talk about it. What are metamers? Metamers are two different spectral power distributions. Again, those are infinite dimensional, that project to the same SML response. Again, those are three-dimensional. Two metamers will appear to have the same color to a human because the brain is receiving the same three SML colors, even if the original spectral power distributions look as graphs completely different, okay? The existence of metamers is critical to color reproduction because it means that we don't have to reproduce the full spectrum of a real world scene. Like we don't have to go into that damselfly scene and at every point reproduce in fine detail, the spectral power distribution at every point. What we need to do is to find a metamer and produce a metamer for that spectral power distribution, and that will appear to a human observer to be exactly the same color. It's an illusion, and it's one that the person, that a human observer will be unable to tell the difference between, okay? So what does it look like? If you look in the real world at the solar disk, you're not really supposed to do that without glasses. So this is just a theoretical example. Okay, there's some spectral power distribution, function of wavelength. If we try to reproduce that on a display and make it look exactly the same, it might look like something like this, okay? It might be this ratio of the primary lights, the RGB values in this case of the pixels in that display. These spectral power distributions don't look anything alike. But to that person, if the color reproduction math has been handled correctly, these two will appear to be the same color. That's the illusion, okay, of metamerism. Metamerism is a really big effect, okay? All of these are metamers too. Look how vastly different they look. These are like three Dirac delta functions, only three spikes in the spectrum. This is sort of more like a bell curve. This one has sort of a big point over here and a hump over there. This one has two sort of, pretty smooth humps. These all will produce the same SML values that go to the brain and will look exactly the same to a human observer. Okay, so metamerism, big effect. It's crucial to color reproduction. Okay, so how does that work? Here's let's get into the math formulation of this in detail now. So let's say you look in the real world at this damselfly. Again, let's pick on this beige background here. There's a target spectral power distribution in this beige background. Of course, S of lambda is different for every position in this image. So we'll do this pixel by pixel, so to speak, across this image. But let's just pick on this beige example as one of those pixels and look at what's happening. What we want to do is on the display, we want to calculate what values to put in, the RGB values, to create an output display spectrum that's going to have the same appearance as that background behind the damselfly. What is the display spectrum that's going to come out? Our degrees of freedom are capital RGB here. They're scalar values representing, like in the color matching experiment, the intensity of the three primary lights in this display, which are the red, green, and blue pixels, or spectrum of R, spectrum of G, and spectrum of B, which are functions of wavelength. Okay, so we get to pick just the RGB scalar values. So the goal is, at every pixel, choose the RGB values for the display so that the output display color matches the appearance of the target color in the world. This is an additive color model, okay? So given the set of primary lights, SR, SG, and SB, we adjust the brightness of those lights and add them together in superposition to produce the output spectral power distribution on the display, okay? So the color now can be described for this display by just these three scalar values at every pixel RGB. Let's look at this, in just a little bit more detail for like a display that we're all familiar with, like on your smartphone. So where is this superposition happening exactly? Oh, remember, if you take a magnifying glass and look at these pixels, they're too small to see individually with the naked eye. But if you look really closely, there's neighboring red, green, and blue areas, the little stripes. Okay, so what are the primary lights? Those primary lights, those dyes in front of those pixels, are the primary red, green, and blue lights in this case. And the intensity that we pass through those red, green, and blue pixels is the RGB value. And then why are they in superposition? They're not overlapping or anything. It's just because they're so small. So for the viewer, apparently they're coming from the same position. You don't have the optical acuity to separate them out. So they just blur together, at least for that one pixel location. That's the way that superposition happens. And for an iPhone, or at least a pretty recent iPhone, these are the actual spectral power distributions with the blue, the green, and the red pixels on those surfaces. So this is S sub B, this is S sub G, and this is S sub R. Okay. Okay, so again, so this is the color reproduction problem. Pick R, pick G, pick B, given those spectral power distributions for red, green, and blue, and given the target. Before talking about the solution mathematically for it, which is going to come pretty quickly, actually, now that we've set it all up, I want to give a pseudo-geometric interpretation. I've sort of been talking about this a lot already, but perhaps this may be helpful conceptually or intuitively. So the high-level intuition or view of this problem is that we're projecting a very high-dimensional vector. That's the spectral power distribution, the wavelength spectrum function, right? Onto a low-dimensional subspace. So a high-dimensional vector projected onto a low-dimensional space. The low-dimensional space in this case is the human visual response to color. That's the SML response. So it's 3D, okay? And one way to sort of picture that, because it's a bit hard to picture infinite dimensional space projected onto 3D, let's cheat a little bit and use a pseudo-geometric interpretation where 3D space will represent this high dimension, and then the low dimension will represent as a plane. Okay, obviously this is a pretty crude analogy, but let's go with it and see if it provides any sort of geometric intuition. So the metamers in this space are things that project onto the low dimensional subspace onto the same point. Okay, so in this case, differences that are perpendicular to the basis vectors of the low dimensional space are not detectable. And we project from the high dimensional space, at least in this picture, vertically down onto the subspace. So here, for example, where are metamers? These 2 points here are metamers. In fact, any point along this projection line that gets down to the same point on the low dimensional subspace, they're all metamers. Okay, they all look exactly the same to the person, even though they're in the high dimensional space. Following or continuing this pseudo-geometric interpretation, when we do color reproduction, we're trying to, the display is also a low-dimensional subspace. It can only produce a low-dimensional subspace of all the possible metamers because it's only the possible spectra that are linear combinations of the display primary lights, right? The red, green, and blue pixels. So it's also a low-dimensional subspace. And in color reproduction, for a given target spectrum S, say that color behind the damselfly in the real world, what we're trying to do is we're trying to find a point in the low dimensional space of the display primaries that is a metamer for S. And therefore, when a person looks at it, will also project onto the same position in the spectral response, low dimensional spectral response function of the eye. So the visual response to both S and S prime being the thing we're going to put on the display, we want both S and S prime to have the same output. Okay. Hopefully that pseudo-geometric interpretation helps a little bit in visualizing infinite space projected into these 3D subspaces. If not, don't worry about it. Or if you have questions about it, come and talk to me about it in office hours, and I'm happy to chat more about it. Okay. Let's talk about color reproduction as linear algebra and get into solving this problem. We're very close now. So the input spectrum S here, what RGB values do we want over here? So we want to solve RGB from input S, and we know it follows linearity. So there's some matrix here that we need to write down that's going to solve this thing. So what is it? What are the pieces of information that we have? We have the spectrum produced by the display given the specific scalar values RGB. we can write it down like this. We have SR, SG, and SB as the output spectra for each of the three pixel types, red, green, blue. We can write those as column vectors here. So they're long vectors, right? Again, it's a function of wavelength. So SR, SG, SB are functions of wavelength, and they're tall here. If you multiply it against a 3x1 RGB vector here, then we would sum these in weighted proportion and get a single column vector that is the spectral power distribution for the display at that location. It's the weighted combination of RGB. It's exactly this function up here, okay? So that's the spectral power distribution of the display. What color does a human perceive when they look at that display is the next question we can ask. We know that too, with any particular input spectral power distribution, the SML response in the eye is given by taking that spectral power distribution and taking the dot product against the S, the M, and the L cone responses, right? R sub S, R sub M, and R sub L. We wrote that down earlier. So the SML response of the eye to the display is given by this top line, and we can expand the display into the components in terms of RGB. So now we have the SML response as a function of the input RGB, and all of these are constants, right? These are constants of the display and constants of the human eye. The next thing is we want this displayed spectrum to be a metamer for the real world target spectrum, right? We want it to look exactly the same to a human being. What's the, so I just copied the previous slide up here. Okay, so SML for the display is equal to RGB times this stuff over here that I described. What is the color perceived for the original real scene spectra? That was S. Remember, this is the real spectral power distribution of the beige background behind the damselfly. It's just taking that S and doing directly the dot product against R sub S, R sub M, R sub L. So this is the real SML response to looking at the real thing in the world. So we want these two to appear the same to a human being. And what does that mean? We just need these two SMLs here to be equal. Okay, we want them to be metamers. Metamers means they're equal. So we just set these two lines equal, and then we're going to solve for RGB as a function of S. Does that make sense? All right, we're almost home free. Let's do it. It's sort of like, it's a bit, it takes up a bit of space to write the algebra down, but it's actually, to write the matrices down rather, but the algebra is almost trivial at this point. So we can write down the left-hand side here equal to the right-hand side. And this implies that we want RGB equal to this thing over here, the inverse of this thing over here multiplied on the left-hand side by, or left multiplied onto this thing on the right-hand side, which is shown here, okay? It's a bit of a mouthful to describe all of it. So I'll just appeal to this solution formula up here by taking the inverse of this part and recognizing that. Question I got asked, great question. Yin asks, is the S vector known to us? Yeah, really good question. In this model, the way I set up the color reproduction problem that we're going to study in this class, yes, the S vector is known to us. Now you might say that's, well, where's it coming from? So you can sort of take it as a given, just the way that we set up this problem in class for our purposes, that it's something that is given to us. But in practice, you might have to measure it or try to estimate it. For example, if you have a camera, you might take it and then you might have 3 projections of that S onto like the color pixels of the camera, and then be able to infer or estimate what the original S is to complete this. But we're sort of taking the simplest version. It's called basic colorimetry, where we're given the full spectral power distribution as an input. Really good question. Thank you. Okay, so this is just copying from the previous slide. The solution form, I'll call it number 1. Let's just observe some of the dimensionalities here. So again, the input spectral power distribution is n by 1, where n is large, right? The number of samples in as a function of wavelength. Call it like 100 for argument's sake. Each of these responses is 3 by, this pack of responses is 3 by n, 3 by 100, say. If you multiply these together, you get a 3 by 1 response. That's going to be the SML response to the eye, of the eye to the real scene. This thing over here, this structure here is n by 3, this matrix. This structure here is 3 by n. So this is a very tall, skinny matrix. This is a very fat, short matrix. If we multiply them together, we're going to end up with a 3 by 3 matrix. one of the ways to think about this is that if we have the original spectral power distribution of the thing we care about in the world, we can find its projection onto the SML response of the eye. And then we can do a transformation, a basis change, essentially a 3x3, into the RGB space of the display that we care about. That's a valid, though somewhat high level, sophisticated linear systems sort of interpretation of this equation, okay? Solution form number 2 is to just sort of write this down in matrix form. So we can call this MSML, we can call this MRGB, MSML is over here and over here. So you can write it sort of in a compact form, just giving names to these vectors over here and write it down in this kind of a format. Kind of out of time, but I wonder if I can just, let me just describe this too. You can observe here by pre-multiplying these matrices together that you can write it down in this format that I've shown over here, just taking dot products between each of the corresponding terms here, R sub S dot product with SMR, R sub S dot with SG, R sub S dot with SB, and then similarly for R sub M in the middle and R sub L in the bottom. This, as you can see, is going to be a 3x3 matrix, okay? So this is the inverse of a 3x3 matrix here. So this is a solution form number 3, where we have a 3x3 matrix here, and then we have this stuff here, which is projecting the spectral power distribution of the thing we care about into the SML space of the eye. Okay. Good. I want to stop here, and we'll pick up from here next week and complete this week's lecture on color reproduction before we move on to talking about image sensors and image processing. Okay. All right. Thanks, everyone. Really good to see you all, and have a good weekend.